Chapter 1, Section 4 Tools and Procedures. Imagine being one of the first people to see living things through a magnifying glass. How surprised you would have been to discover a whole new realm of life. Could there still be other types of life that remain undiscovered today because of the right tools are not available? Scientists select and use equipment which sometimes includes technology such as computers for their investigations. Electronic balances measure the mass of objects with great precision. Microscopes and telescopes make it possible to observe objects that are very small or very far away. With powerful computers, scientists can store and analyze vast collections of data. Biologists have even devised procedures that help them unlock the information stored in the DNA of different organisms. A common measurement system. Because researchers need to replicate each other's experiments, and most experiments involved measurements, scientists need a common system of measurements. Most scientists use the metric system when collecting data and performing experiments. The metric system is a decimal system of measurements whose units are based on certain physical standards and are scaled on multiples of 10. A revised version of the original metric system is called the International System of Units, or SI. The abbreviation SI comes from the French Le Système International of the Units. Because the metric system is based on multiples of 10, it is easy to use. Notice in Figure 1-23 how the basic unit of length, the meter, can be multiplied or divided to measure objects and distances much larger or smaller than a meter. The same process can be used when measuring volume and mass. You can learn more about the metric system in Appendix C. Figure 1-23. Scientists usually use the metric system in their work. This system is easy to use because it is based on multiples. Length uses meters, mass uses grams, volume uses liters, temperature uses degrees Celsius. Analyzing biological data. When scientists collect data, they are often trying to find out whether certain factors changed or remained the same. Often, the simplest way to do that is to record the data in a table and then make a graph. Although you may be able to detect a pattern of change from the data table like the one in Figure 1-24, a graph of the data can make a pattern much easier to recognize and understand. The amount of data produced by biologists today is so huge that no individual can look at more than a tiny fraction of it. To make sense of the data, biologists often turn to computers. For example, computers help determine the structure of molecules. They also allow biologists to search through a DNA molecule, find significant regions of the molecule, and discover how organisms are affected by those regions. At the opposite end of the scale, computers are essential to gathering data by satellite, analyzing the data, and presenting the results. Analysis of satellite data are used to make predictions about complex phenomena such as global climate changes. Figure 124. One way to record data from an experiment is by using data tables. Then the data may be plotted on a graph to make it easier to interpret. Microscopes. When people think of scientific equipment, one of the first tools that comes to mind is the microscope. Microscopes, such as the light microscope in Figure 125, are devices that produce magnified images of structures that are too small to see with the unaided eye. Light microscopes produce magnified images by focusing visible light rays. Electron microscopes produce magnified images by focusing beams of electrons. Since the first microscope was invented, microscope manufacturers have had to deal with two problems. What is the instrument's magnification? That is, how much larger can it make an object appear compared to the object's real size? And how sharp an image can the instrument produce? Figure 1-25. Light microscopes produce magnified images by focusing visible light rays. Light microscopes. The most commonly used microscope is the light microscope. Light microscopes can produce clear images of objects at a magnification of about 1,000 times. Compound light microscopes allow light to pass through the specimen and use two lenses to form an image. Light microscopes make it possible to study dead organisms and their parts, and to observe some tiny organisms and cells while they are still alive. You can refer to Appendix D to learn how to use a compound light microscope. Biologists have developed techniques and procedures to make light microscopes more useful. Chemical stains, also called dyes, can show specific structures in the cell. Fluorescent dyes have been combined with video cameras and computer processing to produce moving three-dimensional images of processes such as cell movement. Electron microscopes. All microscopes are limited in what they reveal. The light microscope cannot produce clear images of objects smaller than 0.2 micrometers, or about 1/15 the diameter of a typical cell. To study even smaller objects, scientists use electron microscopes. Electron microscopes, such as the one shown in Figure 126, use beams of electrons rather than light to produce images. The best electron microscope can produce images almost 1,000 times more detailed than light microscopes can. Biologists use two types of electron microscopes. Transmission electron microscopes, TEMs, shine a beam of electrons through a thin specimen. Scanning electron microscopes, SEMs, scan a narrow beam of electrons back and forth across the surface of a specimen. TEMs can reveal a wealth of detail inside the cell. SEMs or SEMs produce realistic and often dramatic three-dimensional images of the surfaces of objects. Because electron microscopes require a vacuum to operate, samples for both TEM and SEM's work must be preserved and dehydrated before they are placed inside the microscope. This means that living cells cannot be observed with electron microscopes, only with the light microscope. Figure 127 shows images taken with a light microscope a transmission electron microscope, and a scanning electron microscope. Figure 126. This scientist is using an electron microscope to make observations. Electron microscopes produce images by focusing beams of electrons. Figure 127. Observe the images of pollen grains as seen with a light microscope on the left. Transmission electron microscope, the center, the scanning electron microscope on the right. In which image can you see the most detail on the pollen grain surface? Laboratory techniques. Biologists use a variety of techniques to study cells. Two common laboratory techniques are cell culturing and cell fractionation. Cell cultures. To obtain enough material to study, biologists, like the one in Figure 1-28, sometimes place a single cell into a dish containing a nutrient solution. The cell is able to reproduce so that a group of cells, called a cell culture, develops from the single original cell. Cell cultures can be used to test cell responses under controlled conditions, to study interactions between cells, and to select specific cells for further study. Cell fractionation. Suppose you want to study just one part of a cell. How could you separate that one part from the rest of the cell? Biologists often use a technique known as cell fractionation to separate the different cell parts. First, the cells are broken into pieces in a small special blender. Then, the broken cells bits are added to a liquid and placed in a tube. The tube is inserted into a centrifuge, which is an instrument that can spin the tube. Spinning causes the cell parts to separate, with the most dense parts settling near the bottom of the tube. A biologist can then remove the specific part of the cell to be studied by selecting the appropriate layer. Figure 128. This researcher is transferring bacteria to a solid that contains nutrients, which will enable the bacteria to reproduce. How do the results of a cell culture differ from the products of cell fractionation? Working safely in biology. Scientists working in a laboratory or in the field like those in Figure 129 are trained to use safe procedures when carrying out investigations. Laboratory work may involve flames or heating elements, electricity, chemicals, hot liquids, sharp instruments, and breakable glassware. Laboratory or field work may involve contact with living or dead organisms, not just plants, animals, and other living things you can see, but other organisms you cannot see without a microscope. Whenever you work in your biology laboratory, it is important for you to follow safe practices as well. Before performing any activity in this course, study the safety rules in Appendix B. Before you start any activity, read all the steps and make sure that you are understanding the entire procedure, including any safety precautions that must be followed. The single most important rule for your safety is simple. Always follow your teacher's instructions and the textbook directions exactly. If you are in doubt about any part of an activity, always ask your teacher for an explanation. And because you may be in contact with organisms you cannot see, It is essential that you wash your hands thoroughly after every scientific activity. Remember, you are responsible for your own safety and that of your teacher and classmates. If you are handling live animals, you are responsible for their safety as well.